All right, today we're going to be talking about skeletal muscle. Now, you guys learned in anatomy that there are three types of muscle tissue. Skeletal muscle is one of them, but we also have cardiac muscle tissue and smooth muscle. These all have the ability to contract, and that's why they're grouped as muscle tissue. But skeletal muscle is what we're going to be talking about in this unit of study. And of course, it gets its name because it is attached to our skeleton. Now, skeletal muscle cells are long cylindrical cells that have a couple of really noticeable and defining characteristics. So if we were to draw a single muscle cell or muscle fiber, one of the things that we would notice about it right away is that it has these obvious striations on it. Okay, so this skeletal muscle cell has these sort of alternating dark bands and light bands. Here in a little while, we're going to be looking at the source of those bands. Okay. And then, I suppose if we were to label those, all right, we would label these the light bands. Okay. Light bands are also often called eye bands. All right. And then we have the dark bands up here as well. All right. So we have multiple dark bands here, and these are called A bands. Right? Now that I and that A that you see there, those abbreviations, I guess if you will, come from the German words that were first used to describe these by the microscopists that described them. The easiest way to remember which one is the light band, the I band, and which one is the dark band, the A band, is just simply to look at the second letter of each of these words, right? Dark A, light I, and you can remember A and I. It's not why they're named that, but it's an easy way to remember, if nothing else. All right. So skeletal muscle has these obvious striations. Sometimes you'll see in older texts and things like that, you'll sometimes hear skeletal muscle referred to as striated muscle, and you can see why. Now, cardiac muscle also has those striations that are obvious, but we often will reserve that term striated for skeletal muscle. And the other thing that really defines a skeletal muscle cell is that it has multiple nuclei. All right? So we have multiple nuclei as well. There really aren't any other cells in our body that have multiple nuclei. Now, an individual skeletal muscle cell or muscle fiber can be up to a foot or two in length. You know, if you think about some of the longest muscle cells in our body, or longest muscles in our body, you're looking at things like the sartorius, the gracilis, these muscles in our legs. And these muscles, you know, can approach, in tall people, the sartorius can very easily be 2.5 feet long. So the muscle cells, the muscle fibers that make up that muscle are quite long themselves, sometimes up to a foot, maybe even a little bit more in length. So along the length of this cylindrical cell, we have multiple nuclei. All right. Now, the nuclei typically are very obvious when you look at these under the microscope because they have been pushed towards the plasma membrane. Right? If you remember from anatomy, we have kind of a specialized anatomy or vocabulary when we talk about skeletal muscle or any muscle for that matter. Right? So for instance, the plasma membrane of a muscle cell is called the sarcolemma. Remember that prefix sarco means flesh. And we only see that prefix sarco associated with things with muscle. And we're going to see it a couple more times as we talk about skeletal muscle anatomy and physiology. All right? So the plasma membrane of these cells is called the sarcolemma, and the nuclei are pushed towards the sarcolemma, and as a result of that, they are very easily visible. All right? The other thing that we see inside of these muscle cells, when we look inside of these muscle cells, we see a number of organelles that are also long cylindrical organelles called myofibrils. So we have these myofibrils. These are contractile organelles. Very specialized contractile organelles. We don't really see them in any other tissue in our body except for muscle. And we just see that prefix myo. That prefix myo means muscle, and we know for sure that we're talking about something associated with muscles. All right. So the skeletal muscle fiber itself is really just jam-packed full of these myofibrils. And we're going to blow those up here in a couple of minutes, and we're going to take a close look at what one of them looks like. And then, of course, the other thing that we have a lot of in this muscle fiber is we have a lot of organelles called mitochondria, which are small, they don't take up a lot of space, and our skeletal muscle cells are just jam-packed full of them. Right? We talked about ATP synthesis already, and during exercise especially, our skeletal muscle cells need a huge amount of ATP. So it's no surprise to find that the muscle cell itself is just chucky jam full of mitochondria. Right? There's a couple of other organelles that we're going to be talking about as we move through this discussion. I'm not going to try to draw them on here, mostly for the sake of clarity, but another organelle that we're going to be really interested in is something called the sarcoplasmic reticulum. We'll draw it when the time comes, but the sarcoplasmic reticulum is an organelle that we're going to be talking about. Again, we see that prefix sarco, but this is a modified endoplasmic reticulum. It's a modified endoplasmic reticulum, and its job is to store calcium. Okay, so with these things in mind, what I want to do is I want to take a look at one of these myofibrils in a little bit more detail. All right, so let's go ahead and take and blow up one of these myofibrils and see what it looks like. All right. So if we take a look at a myofibril, one of the things that we're going to see is that it looks very similar to the muscle cell that we just looked at. Now remember, there are hundreds or even thousands of these myofibrils inside of an individual muscle cell. And within a muscle, there are many, many muscle cells. So there's kind of this nested anatomy that we're going to see with muscles, that we're going to see with neurons and things like that. Right? So we're seeing this kind of over and over and over, but these things do look very similar. It's important to remember the vocabulary so that we don't confuse what it is that we're looking at. All right? So these myofibrils, like muscle cells, are long cylindrical organelles. Okay? And if we take a look inside of the myofibrils, we're going to find several different types of proteins. Okay. Some of these proteins are contractile. All right, so these are going to allow the muscle to do what we know it does, and that's to contract. So we have contractile proteins. Some of these are regulatory proteins. So in other words, they're going to regulate when the muscle can contract. And then some of these are what we call accessory proteins. Whoops. Right? So we have three different types of proteins. We have contractile proteins, we have regulatory proteins, and we have accessory proteins. Okay. Our contractile proteins are actin and myosin. Whoops. We're going to talk about all of these, so we're just going to list them for right now. All right. Our regulatory proteins are going to be troponin and another one with a similar name called tropomyosin. And then we have some accessory proteins as well. We're going to talk about them when we get to them, but these have two of the coolest names, I think, in the protein world. We have one called Titin, Titin, which is named after one of the moons of, I believe it's Saturn. And then we have a protein called Nebulin. Right? So all in all, we're looking at 6 proteins here, and all of these are going to be really important for muscle function. Let's go ahead and look and see how they are arranged within this myofibril. Okay, within this myofibril. I'm going to make a very large myofibril here just to kind of help to ease my poor art skills. So this is that same myofibril that I had drawn on the previous slide. I'm just going to blow it up a little bit. All right. I'm going to look down here so I can keep my colors straight. All right. So actin is made up of these very small protein subunits. Right? An individual actin molecule. Look down here, sort of at the bottom. Okay, has these individual protein subunits that kind of come together like this. Okay, these protein subunits are called G-actin. Okay, so each one of these individual proteins is a molecule of what we call G-actin. And when they come together like this, the entire thing is called F-actin. Okay, not factin, but F-actin. All right. And what happens is that in a myofibril, we have 2 of these F-actin molecules sort of wrapped around one another. Okay, almost like if you had 2 strings of pearls, what you would do is you would give them that twirl, and they would wrap around one another, and then you would end up with these kind of twisted things like this. So we end up with something that sort of looks like this. Okay, and we have 2 F-actin chains wrapped around one another. We call that a thin filament. All right. So a lot of times we'll use this term actin and we'll use thin filament synonymously. They aren't exactly synonymous, but we often do that sometimes just out of laziness. All right. Now, the other thing that we are concerned with, all right, is we're concerned with myosin. Now, myosin is made up of much heavier proteins than actin. And when we get multiple myosin proteins together, we get what we call thick filaments. So again, we usually end up using myosin and thick filament synonymously with each other. But we have to remember that the thick filament is made up of myosin proteins. And here I've drawn several of them, right? They're all kind of in line with one another. At the end of every single one of those, we have two little heads that sort of come up like this, all right? We're going to talk about the importance of those, but they kind of come up like this. And so at the end of this, when we have a whole bunch of these together, we have a whole bunch of these myosin heads that sort of come up that look sort of like this, all right? So with these two things in mind and how actin and myosin are sort of put together, why don't we go ahead and look at how these thin filaments and these thick filaments interact with each other inside of a myofibril. Okay. So what I'm going to do is I'm going to draw for just a second, and then we're going to kind of describe what we're looking at. Okay. All right, so I'm drawing these myosin, these thick filaments in here, kind of like this. Remember that they all have these little heads that come off of them. I'm not going to take the time to draw them all on, but remember that they are indeed there. All right. And then for the thin filaments, I'm not going to draw each individual G-actin protein like I did here, or even the F-actin with them wrapped around one another. I'm just simply going to draw a thin filament. So our thin filament kind of looks like this. There's a number of them kind of coming like this. You can see that I'm kind of drawing them overlapping with our thick filaments. So we end up with something that sort of looks like that. All right. This. All right, and then we kind of keep going with this. We would have our thick filaments over here on this end. All right, go from there. Going this way, and so on and so forth. Hopefully you can see at this point that I'm drawing a pattern that is simply going to repeat itself, right? Kind of like you were in elementary school and I asked you to pick out the pattern. You can see that there's a pattern here that's sort of repeating itself, all right? So we have our thick filaments, we have our thin filaments. We see that there is some overlap there, all right? What I want to do is I want to focus on a portion of this. Okay, I want to focus on the portion from here to here, all right? The proteins that hold these thin filaments in place, this sort of zigzaggedy line that I'm drawing right here, or tracing right here, this is called a Z-disc. So we have a Z-disc that kind of holds our thin filaments in place within the myofibril, all right? We have another one right here. And the distance from one Z-disc to the other Z-disc, this distance that we have right here, is called a sarcomere. Okay? So the myofibril itself is made up of these repeating sarcomeres, right? I have one here, I could go down here, and it would repeat all the way down the length of the myofibril. You know, for one of these that is a foot long or a foot and a half long or something like that, we would be looking at hundreds or even thousands of these sarcomeres within one of these myofibrils. But this is essentially what it looks like. All right. Now, because the myosin filaments are a much heavier protein than the actin filaments, what we end up with is we end up with alternating light and dark bands, right? So if I were to sort of draw right here, what we would see here is that this is an A band, right? This is our A band that we see right here. The distance that we have then between here and this next beginning of this A band, this is our I band. Right? So just like with the skeletal muscle fiber that I drew here at the beginning of this lecture, just to remind you of that, we saw those alternating light bands and dark bands right here. When we look at one of the individual myofibrils, we see those alternating light and dark bands, right? And we can see what they're coming from. They're coming from the arrangement of these proteins within the myofibril, all right? So when we look at the muscle cells, we can see that that is due to the arrangement of these proteins. You have thousands of these proteins in the cell, and that becomes visible when we look at this underneath the microscope. All right, so we have the A bands, we have the I bands, we have the Z discs. We also have the line where the thick filaments are being held in place. This is called the M line. All right, so we have the M line right here. Right, so we have the Z discs, the M line, we have our A bands, we have our I bands. All right. There's one other sort of portion of this that I want to point out because it's going to become important here as we kind of move through this. You'll notice that within this individual dark band that I've drawn for you right here, there is this sort of portion of it where the thin filaments and the thick filaments overlap. But at the same time, there is a portion of that dark band, basically from here over to here, where there is no overlap, right? It's only myosin protein. This is called the H zone. All right? So we have the H zone here as well, and we're going to return to that probably in the next slide as we kind of go through this. All right? So what we have here is we have this arrangement of these proteins. All right? And I'm going to give you a little bit of a spoiler alert, but I'm not going to describe a whole lot of mechanism to you quite at this point. And I'm going to tell you that when the muscle shortens, these filaments are going to slide past each other. And one of the things that we're going to do is we're going to be looking at the mechanism of that. Right? But muscle doesn't just have the ability to shorten. Muscle also has the ability to stretch, to get longer, right? You know, when you wake up in the morning, you stretch. When you go running or play basketball or something like that, you stretch as well. And unless you really screw things up and stretch too far, your muscle goes back to its original length, right? So we have the ability to contract our muscle, right? But we also have the ability to stretch that muscle out. And then it also goes back to its original length. So not only is muscle contractile, it's also extensible, meaning that we can stretch it out, and it's elastic, meaning that it should return to its original length. So those two characteristics, extensibility and elasticity, are where those accessory proteins, titin and nebulin, come in. So I just want to draw those in here. We're not going to talk a whole lot about them, but I want you to know where they sort of go, right? So titin is this really quite large protein that essentially anchors the end of these thin filaments to the M line. So we kind of see it in here like this. And I'm drawing it sort of to look like a spring because that's essentially what it does. All right, it kind of goes on the other side here as well. When we stretch one of these sarcomeres out, when you stretch a muscle, all right, when you stretch a muscle, what you're doing is you're separating these sarcomeres like this. And then when you relax, you stop that stretch, you hope that muscle fiber goes back to its original length, right? These springs, this titin, sort of kind of pull it back together like that, all right? And then nebulin is found in here as well. Nebulin sort of keeps everything arranged. It kind of runs down through here like this and just sort of keeps everything arranged the way it's supposed to stay arranged. Because as you can imagine, this is a fairly complex series of wires, if you will, and proteins, and it's helpful to keep things where they're at. So that's where we find titin and nebulin. All right, titin is involved with the extensibility and elasticity of the muscle, but we're not going to talk about it a whole lot more than that. What I really want to focus on here are the arrangement of these thin filaments and these thick filaments. So muscles do contract. One of the things I'm going to do is I'm going to redraw this on the next slide, and I want to illustrate this for you. So what I'm going to do is I'm going to draw a relaxed sarcomere, and I'm going to draw a contracted sarcomere. Now, bear with me for a minute while I try to make these about the same as one another. All right. We have a relaxed sarcomere. So I'm going to actually draw my thick filaments first. So we have our myosin. So those are thick filaments that we had, right? And draw a second one down here. And try to make it the same as the one that I've drawn up above. So if they don't look exactly the same, humor me a little bit, but this is what we have, right? And then we have our thin filaments. I'm going to draw them here first. So we're going to draw our sarcomere again. Something like that anyway. Looks like I got it right. All right. So let's go ahead and label some things. All right. The first thing that we have is we have our entire sarcomere, right? We have our entire sarcomere here. All right, we're going to call that A. And we're going to write over here that A equals the sarcomere. All right. And then we have part, I didn't draw the entire thing, right, because I only drew one sarcomere. But from here over to here, we're going to call that B. And B is going to be 1/2 of an I band. Right? We have right here, C, we have an A band. Capital A just to stay consistent here. And then we have this one section here that I drew for you guys on the last slide as well. Remember, that's our H zone. We're going to call that D. Let's say this is our H zone. Okay? So those are what we have in this relaxed sarcomere, and that's identical to what I had on the last slide as well. What I want to do now is I want to go ahead and draw a contracted sarcomere. Right? So early anatomists were basically comparing relaxed muscle cells with contracted muscle cells underneath the microscope, and these are the things that they first noticed. All right? So this is a contracted sarcomere here, and I'm going to really exaggerate it, hopefully, to sort of drive this point home for you guys. Right? So there's our contracted sarcomere. And I didn't do it perfectly, but I think I'm going to be able to drive my point home. All right? First thing we want to look at in terms of comparison is we want to look at our sarcomere itself. Has the sarcomere gotten shorter or has it gotten longer? It's gotten shorter, right? So the sarcomere has shortened. And because the muscle is essentially made up of repeating sarcomeres. We know muscles get shorter. It makes perfect sense that the sarcomere would have gotten shorter.